ask your question directly, you can raise your hand and Greg will make you a panelist and you can pose your questions to Nick directly. All right, so without further ado, join me in welcoming Nick.
Thank you. Thank you for the nice introduction. Let me get this going here. All right. Does that look good?
Yeah. Yes.
Perfect. Yeah. Hold on. Let me get this out of the way here. Sorry about that. Oh, yeah. Thanks for that introduction. Thanks for inviting me to present this really great seminar series. I've been able to attend quite a few of them, and I think they're fantastic. So really honored to be invited to give one myself. And for anybody that knows me, I guess maybe not that many people since I just started the lab, this talk may come as a bit of a surprise. I'm going to talk about the urinary bladder, which I don't think many people know. I actually do any work on that. And in this title slide, you can see two videos of the same urinary bladder under normoxic or normal oxygen conditions. You see lots of calcium activity going on in that bladder. And then to the right, you see a bladder that's been exposed to acute hypoxic conditions, and that's actually a video, but it's really not moving. And I hope to explain this, how this occurs clearly to you as throughout this talk. And as was said just a second ago, I did just start my lab, so I'm going to take another moment to shamelessly plug what we're doing here in Vermont and talk about the other major component of the lab, which is the brain and retinal vasculature, with a particular interest in capillary pericytes, which sit on all these tiny blood vessels within the brain and retina, and how these pericytes can maybe sense environmental conditions, regulate blood flow, and regulate vascular integrity, with a large component focusing on how ion channels may mediate these responses. But as you'll see today, the lab has a deep interest also in like receptor to ion channel signaling in diverse types of smooth muscles, such as arteries and veins, in different bladder tissue. So today I'm quite pleased to tell you sort of a full story about the urinary bladder, which is came from a very collaborative project with Jerry Herrera, who's also here in the Department of Pharmacology. All right, so when we think of the bladder, it can be kind of generally defined as this big hollow muscular organ that stores urine and voids urine that's produced by the kidneys. And we take a closer look at the bladder, you see it's made of three principal layers, where the inner layer of the bladder, or this part that comes in contact with the urine, is made out of these cells called urothelial cells, and provides a nice barrier to keep to protect the rest of the bladder from this urine environment. Then when you look in the middle layer, you see this maybe less well-characterized layer, the interstitial layer, filled with interstitial cells, blood vessels, nerves. And then this outer layer is this really thick muscular layer filled with smooth muscle cells. And that's probably what most people think of when they think of the bladder, is this big muscular layer that's all around the bladder. This is called the detrusor layer. So if I say detrusor, I'm just talking about the big muscular layer around the bladder. And since this is the muscular layer, it's the part that produces all the contractility within the bladder. So these three specialized layers, it seems like they have a pretty simple task to stay relaxed in order to store urine. And then at some point, they need to be able to sense fullness and send that cue to the central nervous system that the bladder is full. And then you get this brief but forceful contraction to void urine. And then the bladder should go back to a relaxed state to store urine. It seems simple, and actually that dogma still persists today in the clinical and integrative physiology literature. And so this figure sort of shows that, sort of suggesting that the bladder is completely relaxed during filling, that the detrusor is completely relaxed while you start getting the first sensations that you need to void. And then only when you have this cue to void through the parasympathetic system does the smooth muscle layer around the bladder become active and squeeze and cause a voiding event. And then right after that occurs, we go back to a condition where the bladder smooth muscle cells are all relaxed and filling. But that's actually not what's going on. the bladder is not relaxed during filling. It's actually remarkably active. And this is a recording from a healthy human bladder where here we see a voiding event where the detrusor pressure goes up and the intravascular pressure goes up, and we have flow or a voiding event. But before this event, we have lots of detrusor activity, lots of pressure events going on in the bladder. And these events are driven by the bladder constricting, the detrusor layer driving these non-voiding pressure events. And Tom Hepner and Nathan Taikaki and colleagues from the Nelson Lab, I guess almost 10 years ago now, they did a really nice job demonstrating the importance of these non-voiding contractions. So not just voiding, but these large contractions that are occurring while the bladder is filling. And here they used an isolated mouse bladder with an attached afferent nerve activity recording. So they can fill the mouse bladder, and they see as they fill the bladder with saline, they're just filling the bladder up. You see these transient contractions starting to occur, and then right in line with the beginning of these transient contractions, we see a burst of afferent nerve activity. So the bladder is it's transmitting nervous system activity, nerve signals back up to the central nervous system. And I think the key of this paper is they showed that the transient contractions are what's really driving these bursts in afferent nerve activity. So it's really smooth muscle cell activity. It's tied to these afferent nerve bursts, giving the brain information about how full the bladder may be. But I also wanted to take a moment on this to point out maybe the unique characteristics of the bladder's smooth muscle cell action potential. So unlike a nerve or a skeletal or cardiac muscle, the upstroke of a bladder smooth muscle cell action potential is driven mostly by voltage-dependent calcium channel activity, and then the repolarization phase, or this downstroke, is driven mostly by activity of these large conductance calcium-activated potassium channels, or these BK channels. So you have action potentials going off in the bladder smooth muscle cells, driving these transient pressure events, which drive the sensation of fullness within the bladder. So the main point I'm trying to make with this slide is that, even when the bladder is not voiding, this ongoing muscular activity is really key for bladder function. And I put that here. I just hope that I can communicate that one point, that the smooth muscle activity within the bladder is really important during filling and during the voiding phases. It's not just relaxed doing nothing during the filling phases. So why does this matter? So during this normal filling and sensing of bladder, during normal filling and the bladder sensing this filling, you should have normal voiding frequency and volume. So this shouldn't disrupt, your normal daily living or quality of life. It should be spaced out and painless. However, there are many types of bladder pathology that result in a big range of symptoms that can vary from being inconvenient to even debilitating. And this includes incontinence, overactive bladder conditions, or having lots of frequent small voids, underactive bladder, which is probably a worse problem than overactive bladder, leading to the bladder just not being able to work anymore, increased urge frequency, and dysfunctional voiding. So these problems, they may seem minor to people that don't experience them, but I think for those that have these issues, and especially if they are accompanied with pain, are quite detrimental to quality of life. And the aspect of this that really interests me and sort of brought me into the bladder world a little bit was that during normal filling and voiding, the bladder will experience transient hypoxic conditions where the muscle activity is the greatest. So during transient bladder contractions, we have areas within the bladder that become briefly hypoxic, or no blood is being delivered to those regions. And then also when you have a voiding event, the bladder contracts down so forcefully that the whole bladder becomes hypoxic. And then that seems to be normal transient hypoxia that's resolved. But when we talk about the pathophysiology that occurs causing over, under active bladder and a whole host of other bladder conditions, this transient hypoxia seems to develop into more chronic ischemic hypoxic conditions, and chronic hypoxia seems to be a putative underlying mechanism for almost all of the pathological bladder conditions. Water. So in other excitable tissues like the heart and the brain, both physiological and pathological hypoxia are linked tightly with local adenosine levels, where if the tissue experiences dips in oxygen, you'll have a rapid and substantial increase in extracellular adenosine concentration. And this is met with adenosine receptor-mediated signaling and essentially an adaptive response to that hypoxia. And in smooth muscle cells, at least most of the smooth muscle cells that this has been characterized in, the adenosine signaling cascade occurs through stimulation of A2A or A2B receptors, or this family of A2 GS protein coupled receptors, leading to the production of PKA, and PK playing a key role downstream to mediate smooth muscle cell responses to adenosine. Okay, so with all that said, and because hypoxic conditions seem to occur in normal and pathological bladder conditions, and we actually know very little about adenosine signaling and hypoxic signaling in bladder tissue, I mean, the bladder in general is an understudied organ. We wondered, how is adenosine and hypoxia sensed by the bladder, if it's sensed by the bladder, and are these two things linked in the bladder tissue? So we first addressed this question using a similar approach to the other paper I was talking about, this whole ex vivo mouse bladder, where we could take the bladder out of the mouse, we can cannulate the bladder, we could fill it up to a normal volume and a normal pressure where they produce these nice, reliable transient pressure events, those events that seem to be really key even in the pre-voiding phase. And also it's a bath perfuse, so temperature, pH, and all those other factors are regulated nicely. And in many of these experiments, we also were able to use mice that had a genetically encoded calcium indicator, this GCAMP6F, specifically in smooth muscle cells. And so we could observe not only these transient pressure events, we could also observe how the spatial and temporal aspects of muscle activity are driving these transient pressure events. We just did this using basic wide field microscopy. And so you could see in this video that these transient pressure events are driven by these cascading neighboring action potentials occurring across the surface of the urinary bladder. So the first question was pretty simple to address. What is the effect of putting exogenous adenosine on the bladder? How does that affect bladder activity? And so you can see these pressure traces, and these clearly show the dramatic silencing effect of adenosine, where here we use this analog 2-chloroadenosine, which throughout the talk would just be called CADO. This is just a stable adenosine analog, so we kind of fix the concentration without the adenosine being broken down in the bath. And then when we incubate the bladder with it's actually a quite nice drug, PSP603, which is a specific A2B receptor antagonist, we don't see much change in bladder activity just by incubating with this drug alone. But then when we subsequently add adenosine back into the bath, we see that this silencing effect is now lost, and we have this normal bladder activity, suggesting that the effects of adenosine are almost solely through the A2B receptor. And other groups have showed that the A2A receptor, and we also did this as well, it doesn't seem like the A2A receptor is doing much in bladder smooth muscle cells. So it seems to be all A2B, and they have similar downstream signaling. And then we also added forskolin in the presence of the A2B blockade and with adenosine on, to show that if you just stimulate cyclic AMP and PKA directly, we can go back to this silent bladder, no bladder activity. And these data here are summarized here, looking at bladder event amplitude and bladder event frequency, where we're just comparing these things to baseline. And as shown in the traces, adenosine has a potent silencing effect, forskolin as well. But when we block the A2B receptor, doesn't seem like adenosine's doing much. So as I mentioned earlier, we paired a lot of these experiments using this fluorescent calcium imaging of GCamp. So in the same experiments that generated those pressure events, we could also image the prevalence and coordination of smooth muscle cell action potentials across the surface of the bladder. So the nice thing here is, the pressure traces are nice because they tell us a lot about bladder, the smooth muscle cell contractility, but this tells us how it's coordinated across the tissue. So now we have some spatial temporal information. And to do this, we actually utilize software developed by Grant and Hedig, where we're able to extract calcium events, which you see here in the middle panel, and then compile those on top of each other to look at where within the bladder the most calcium activity is occurring. And then, as we saw with the pressure events, when we expose the bladder to adenosine, we see a dramatic loss in total bladder smooth muscle cell action potentials across the entire surface of the bladder. So loss of total prevalence of action potentials and the coordination of these action potential events across the bladder. And this is quantified here where you can kind of see the spatial temporal map of total calcium activity. And then we also did this nice analysis showing something called coincidence, which is not only how often calcium events occur in bladder smooth muscle cell bundles, but how often they are to cause a neighboring bundle to fire. And we think that this neighboring muscle cell firing is really important to coordinate these transient pressure events because they need to be coordinated in some way. And so adenosine, the effects were so dramatic, it wasn't a real surprise that both of these things were heavily affected, both total prevalence of calcium events and the coordination of these events across the surface. And then, of course, forskolin completely shut down the calcium events across the bladder. So this potent effect of downstream adenosine receptors in the bladder smooth muscle cells, it got me pretty excited because at the same time maybe a little bit before this, we were working out that adenosine had dramatic hyperpolarizing effects within the brain capillaries. We published this paper a few years ago that the effects of adenosine within the brain capillaries, the pericytes and the endothelial cells, was transduced through activation of vascular KTP channels, which are highly sensitive to PKA. So then we asked the question of, what is the hyperpolarizing ion channel downstream of A2B receptors within the bladder smooth muscle cells, and sort of naively assumed that it would be the KATP channel in bladder smooth muscle cells as well. And so what we first did was just, functionally characterize that the KATP channels are in the bladder smooth muscle cells. So here we have those normal transient pressure events with the full bladder being recorded. And then we could add pinacidil, which is a potent and specific synthetic activator of the KATP channel. And this completely silenced bladder activity. So we knew the KATP channel was there. The activation of this potassium channel caused sufficient hyperpolarization to pretty much remove these contractile events in the bladder. And then we put glibenclamide on, which is a blocker of the KATP channel. And we see that there's no real difference between baseline and glibenclamide conditions. And then we added pinacidil on top of glibenclamide, showing that glibenclamide is a good blocker. The pharmacology is working the way we'd expect, and that transient pressure events are unaffected by pinacidil when we block the KATP channel. And then these traces are just summarized here when we look at pressure event amplitude and frequency. So no surprise that pinacidil has a huge relaxing effect on the bladder. But to our surprise, we saw that blocking the KATP channel did not really do anything. We thought that if adenosine was activating the KATP channel through PKA, that blocking this channel with glibenclamide and then adding adenosine back into the system, that we would still see these transient pressure events. And so we blocked using this 10 micromolar glibenclamide and then added adenosine back to the system. And the bladder was again completely relaxed in response to adenosine. So it's interesting that the bladder smooth muscle cells contain the KATP channel. It seems they contain the 6.1 and SIR2 subunits, but they don't seem to be sensitive to adenosine receptor production of PKA, at least in our hands. But as you see in the schematic, PKA is not only activating this one potassium channel, the ATP sensitive potassium channel, it has a large effect also on this other ion channel that plays a huge role in the bladder's smooth muscle cell action potential, this large conductance calcium activated potassium channel, the BK channel. So we tried to explore whether this was what was going on downstream of adenosine. And since BK channel plays a huge role in repolarizing the action potential, when we block the BK channel with Paxilin, we see a huge enhancement of bladder activity. But then when we subsequently add adenosine to the bladder, we don't see any sort of relaxation effect at all. So we figured, this is enhanced, but we figured if adenosine was working through some other inhibitory target, that we'd see some reduction of this contractile forces, and we didn't see that at all with when we pre-incubated with the BK channel blocker Paxilin. So this sort of gave us the suggestion that maybe BK channel is what's being activated by PKA downstream of adenosine, but this is pretty artificially enhanced because we're messing with the action potential of the smooth muscle cell. So then we turned to using electrophysiology to see what the effects of adenosine are on whole cell currents, in particular the BK channel. So here we used isolated bladder smooth muscle cells. So with the same bladders we were using before, we could use enzymatic digestion to get single bladder smooth muscle cells. We could perform whole cell patch clamp recordings with physiological potassium concentrations. And then we did a -70 to +70 millivolt voltage step protocol. And you see these large outward currents in response to this protocol in these conditions. And then when we add adenosine to the bath for about 20 minutes, we see that these outward currents are hugely enhanced, and that these enhanced currents are sensitive to the BK channel blocker Paxilin. However, when we take these smooth muscle cells and then pre-incubate them in the A2B channel blocker, this PSB603, and then subsequently add adenosine, we don't see much enhancement of the current at all, and these currents are still sensitive to Paxilin, or the remaining current is still sensitive to Paxilin, so there is some residual BK channel activity. And so we see that this adenosine-induced Paxilin-sensitive current is blocked by blocking the A2B channel. So further evidence that adenosine seems to be activating the BK channels in these smooth muscle cells. And I know this is the EpIC seminar, and it's lots of electrophysiology and structure and function, but I have to give the disclaimer, this is my only electrophysiology slide, so apologies for that. Okay, so here's this. Oh, sorry. So it seems clear that adenosine has potent relaxing effects on bladder smooth muscle cells. And I spoke earlier that hypoxia seems to be linked to adenosine levels in other excitatory tissues. So what is the effect of hypoxia or low oxygen conditions on the whole bladder? And are the hypoxia effect tied to the adenosine signaling that we've already characterized linked to BK? So to test this, we took maybe a bit of a dramatic approach, but I felt that it was nicely reproducible, where we could actually just scavenge oxygen using an oxygen scavenger. So we get extreme hypoxia or almost anoxia by adding sodium sulfite, an oxygen scavenger, where normal PO2, here these are just oxygen levels in the bath measured as percentage. So you bubble with 20% oxygen. The solution that hits the bladder is also 20% oxygen, so pretty much right with that. And then when you bubble with nitrogen and CO2 to maintain pH, we see 0.1, 0.2% oxygen in the bath. And this corresponds to about 150 millimeters of mercury partial pressure of oxygen and about 1 millimeter mercury partial pressure of oxygen. So pretty extreme hypoxia, but it was a nice way to test mechanisms, we felt. And so when we expose the bladder to hypoxia, here's normal bladder activity, these transient bladder events, we see a complete silencing of the bladder. And again, when we pre-treat with this A2B blocker, looks a lot like the control conditions, and then we subsequently expose the bladder to these hypoxic conditions, we see that this silencing effect is completely blocked. So it looks like the acute hypoxic effects of silencing the bladder are mediated almost solely through the A2B receptor. And this is quantified here in the same way, where we see pressure event amplitude completely relaxed due to hypoxia exposure, frequency goes down, and blocking A2B receptors seems to completely restore bladder activity in the presence of hypoxia. We also did the calcium imaging as well, and missing from this video is the control condition, but you can see here that hypoxia has a potent effect of these action potentials occurring across the bladder surface. Pre-incubation with A2B receptor antagonists looks a lot like control conditions, and then when we expose the bladder to acute severe hypoxia in the presence of the A2B, we see lots of action potentials going off across the bladder. And that's quantified here using prevalence and coincidence as a readout. And it looks a lot like adenosine. So no surprise there. It seems to be the hypoxia and the adenosine signaling are kind of go hand in hand in this tissue. So the summary so far, I hope you could take away from this, is that, acute stimulation of smooth muscle A2B receptors is potently silences intrinsic bladder activity through the stimulation, or at least a large part of it's through the BK channels. And this could be done either directly by adding exogenous adenosine like we did, or by exposing the tissue to acute low oxygen conditions. And this could be, all the details of this were recently published in the Journal of Physiology. And so you could, if you want to read more about this whole signaling pathway, that's could be found here, and we're quite proud of that work. It was published in October. But the outstanding question is, where is hypoxia being sensed? So we can expose the bladder to hypoxia. It seems like it stimulates adenosine receptors, but what's going on beyond that? In that paper, we didn't really get into details there, and that's sort of what we're working on now. So here's some unpublished kind of preliminary data to sort of extend the story a little bit further. So within the bladder literature, it's suggested that the urothelium can actually release ATP, and that this ATP within this interstitial space can be hydrolyzed into adenosine, and that or that even adenosine could be released directly from these urothelial cells. Also, kind of interesting anatomical setup where most of the capillary networks within the bladder are proximal to the urothelial cell layer, meaning that it might be a good site for sensing changes in substrate availability like oxygen, glucose, or just metabolic dysfunction in general. So we utilize that same mouse model we had, this MYH11 GCAMP6F model, so an inducible smooth muscle GCAMP. And what we could do is we developed this hemi-prep. So we cut the bladder in half. And so with half of the tissue, we can pin it down with the urothelium intact. So just pinning down half the bladder as it is. And then the other half, we could surgically remove the urothelium and then also pin that down into a perfusion chamber. And when you pin this out tight, you get the same, kind of intrinsic calcium activity occurring across the bladder sheets. And so it makes for a nice way to read calcium activity in response to different conditions with and without the urothelium intact. And using that same prevalence mapping, we see under baseline conditions, the calcium activity across both sides of the bladder looks pretty similar. Like there's plenty of ongoing calcium activity, action potentials in the smooth muscle cells. And then when we expose the preparation to hypoxia, it looks like only the half that has urothelium intact is subject to the silencing effect of hypoxia, kind of indicating that the urothelium is either sensing it or needed to transduce this signal upon the smooth muscle cells in response to low oxygen conditions. And this is a bit summarized here. This is early days for this work, but we really think that the urothelial cell layer is playing a key role in sensing oxygen levels and other substrate availability within the bladder, and signaling to the detrusor smooth muscle cell layer accordingly. So here you see only when you have the urothelium intact, you have this loss of robust calcium activity or this intrinsic smooth muscle cell activity within the bladder. If we block the A2B receptor with this drug, this PSP603, we see that the hypoxia effect is also blocked when you have the urothelium and without the urothelium. So putting it all together, this is obviously kind of a bit more fundamental to what's the molecular mechanisms that mediate hypoxic effects within the bladder. But we hope that this could be potentially utilized to maybe ameliorate some of the pathology seen in many of these bladder diseases. So the summary for what I'd shown you is that adenosine signaling has a potent relaxing effect on bladder smooth muscle cells, that the A2B receptor signaling can stimulate BK channel activity, and that BK channel activity, increased BK channel activity causes bladder relaxation. The surprising effect, at least to me, was that there was no involvement of the KTP channels, even though they're there. We figured that they should be sensitive to PK, but they weren't with this adenosine paradigm. And then acute severe hypoxia stimulates A2B receptor signaling to relax the bladder. That might be a compensatory mechanism. When you don't have a lot of oxygen, you probably don't want a lot of muscle activity going to that region. And that the urothelium is required to mediate this hypoxia-induced bladder relaxation. So it seems like the tissue layers are working together to mediate this hypoxic response. I'd really like to thank everybody that showed up. I know it's the holidays, and I appreciate your attention and being here. I got to thank Jerry, who was, a really great person to collaborate with. He taught me a lot about the bladder and got me excited about these bladder smooth muscle cells. A lot of people in Mark Nelson's lab helped with this project too. Grant Hennig did the calcium analysis. A really talented grad student, Jason Rango, did a lot of the experiments, especially with the ones that we were scratching our heads about KTP not being involved. Tom Hepner, Maria Sancho, and Saul Huerta de la Cruz. Here's the people in my lab. We are very lucky to be in a new building. Doesn't look like this now. It's completely white outside. And thanks to the funding sources. I'm happy to take any questions.
Thank you, Nick. So everyone, if you have questions, please type them in the Q&A box, or you can raise your hand and ask your question directly. So we've got the first question in from Colin Nichols. He says, sorry, I missed the beginning, but very interesting what I saw. The urothelium requirement for hypoxia sensing, is it O2 per se that is being sensed or metabolic suppression? Can you mimic the effects with metabolic blockers?
Yeah, hi, Colin. Great question. And I guess the general answer is I don't know yet. We did a rather crude approach with just getting rid of oxygen. I don't know if the oxygen is being sensed. What's interesting is that when we block the A2B receptor in those conditions, everything seems fine. So when we remove oxygen, at least for a short period of time, the bladder smooth muscle cells and the seem like they're doing fine. What's going on with the urothelium, I don't know. It is a good question, and perhaps we should try something like that. It'd be interesting to target the urothelium specifically too. Yeah, thanks for the question.
A comment from David Colboon says, it's good to hear some useful physiology. Thank you.
Well, thanks. Yeah,
Okay.
sorry about the lack of EFIS data, but um, yeah.
All right. So we've got some live questions. Connie, would you like to go?
Yeah, thanks, Layla. Nick, that was lovely. Really nice data. Thanks for the talk. And thanks for ruling out KATP. That gives me one less thing to worry about.
Yeah.
Um, I was looking at your videos. I don't know if I missed this, but it looked in some of the videos like there were regions that were driving the wave of calcium response. So are there like
Mhm.
particularly hyperexcitable cells? And does that reflect an intrinsic excitability of the smooth muscle, or is there some anatomical difference with the coupling with the urothelium or the underlying nerves?
Yeah, that's a really good question. It's an interesting question. I don't know. We haven't studied it in detail to give you like a nice empirical answer, but from the look of it, it does look like in the bladder and in other like hollow organs like the gut, there are sites that seem to be like initiators of these calcium things that kind of spread and propagate across the tissue. Um, we're sort of limited with this wide field imaging. You saw in the title slide, there was like multiple views of the bladder. So I think we're mainly the bladder people here are interested in addressing that question. Are there sites like that are driving like kind of like sources for like pacemaking within the tissue? Uh, but you know, very, very, very non-scientifically, it looks like there are sites that drive it, but what's causing it, we don't know. There is some uh, interest in that there are pacemaker cells within the bladder, these interstitial pacemaker cells that may be driving these calcium, these initial calcium bundles to fire and then cause the neighboring firing to occur. And that certainly occurs in the gut, but so far in the bladder, it's unclear what's going on there.
Great, lovely talk. Thank you.
Yeah, thank you.
Our next live question, Marcel, would you like to go?
Hi, Nicholas. Great talk. So in the beginning, you were sort of mentioning the importance of these sort of pre-voiding contractions as opposed to the canonical textbook depiction of how the bladder works. Is there evidence that they're important? I mean, clearly the muscle is doing something, but without those, can you not void later or?
Yeah, I mean it's hard to do cause and effect, at least in the especially in the human. But it seems like a lot of they do this technique called cystometry where they could fill the bladder and look at um, the occurrence of these non-voiding bladder contractions. And in a whole host of bladder diseases, it looks like they're really messed up those non-voiding contractions. Whether or not that's completely related to the ability of the bladder to sense and signal, it I would say the best evidence is some of those preclinical or translational studies from the Nelson group that these bursts of muscle activity are tied to the burst in afferent nerve activity, and that if you were to silence these pre-voiding contractions, you'd probably have less perception of fullness. But that's a really hard question to ask with the tools that we have right now, for sure. Um, so I guess the killer experiment would be to like somehow figure out how to get rid of those non-voiding contractions, but preserve the parasympathetic contraction that occurs with voiding, and see if the normal daily rhythm of storing and voiding is disrupted. And we haven't gotten to that point yet. There's a lot of work to be done in the bladder. I think it's a remarkable organ, and we're just like scratching the surface for sure.
Our next question in the chat. So Kevin Foskett asks, what is the mechanism of nucleotide release?
Yeah, great question. We don't know. We did a few experiments we targeted those endonucleotidases, and by inhibiting those, it looked like the effects of hypoxia were well, a little bit suppressed, but the drugs to inhibit those enzymes weren't great anyways. So that sort of hinted that it's maybe ATP release from the urothelium, and how the ATP is being released. I don't know, maybe through the vesicular release of ATP or connexins. I feel bad keep giving the same answer that we don't understand very much about the bladder, but the urothelial cells seem like they could potentially release ATP through some groups suggest that it can happen through these pannexin channels. Other groups say it occurs through vesicular release of ATP. I honestly don't know. Or you could just have release of adenosine directly, because those experiments never really were too convincing to me in the first place. So unsatisfactory answers so far, but we don't really know how ATP is being released from the urothelium, or if it's even being released from the urothelium. Another possibility is that it's being released from the smooth muscle cells, and that the urothelium contains the enzymes to break down ATP and mediate that adenosine response. We're really agnostic to anything right now. But thanks for the question.
The next question comes from an anonymous attendee. Did you test less severe hypoxia, so say 10% O2 as well? Do you think vascular CMCs may exhibit similar effects as bladder CMC?
Yeah, when we were doing this paper, one of the reviewers was critical of the oxygen levels, but I would say that the non-physiological thing is the 20% oxygen. That's a different point altogether. But to answer the question directly, we did try different concentrations of sodium sulfite, which give you like a graded chelation of oxygen. And so even with really low amounts of sodium sulfite, which kept kind of half the bath concentration of oxygen, we still saw a suppression of bladder activity. So it's definitely occurring. It's just you didn't get these big striking, whopping effects. So studying the mechanisms seemed a little bit more difficult with the partial inhibition levels. And then we also just bubbled with just pure nitrogen, and we see that also silences the bladder. So it doesn't seem to be an effect of sodium sulfite per se.
Next question comes from Raul Esteves Povedano. Why have you used 2-chloroadenosine and not an A2B agonist?
Oh yeah, so the bladder group here, it has a deep interest in ATP signaling within the bladder as well. So when you add ATP to the bladder, it's excitable. But they mainly used the stable ATP. So maybe trying ATP would be interesting. I would expect that you'd see an initial excitatory response from the bladder smooth muscle cells, but you're right. It would be interesting to see if that ATP gets broken down into adenosine. We haven't tried that. Maybe we should try that.
Follow-up question from Kevin Foskett. Is the vasculature intact in the urothelium denuded preparation?
Yeah, that's a great question. I actually haven't thought of that. When we peel off the urothelium, it's a pretty crude thing to do. We think that we're even peeling off some of the interstitium. So I would guess that we're peeling off a lot of the capillary network too when we peel off the urothelium. Is he thinking that maybe the vasculature may be sending or signaling within the tissue? Sorry, it's hard to ask a question back to a text question, but
and we have Paul Wagner, who says, how does the A2B signaling act on the BK channel?
good question. We have a long discussion in the paper about that, and we'd love to follow this up with more detailed mechanism. So PKA and smooth muscle cell, it may be phosphorylating the BK channel directly. There's some evidence that in some tissue types that can cause activation of the channel. I forget the exact site, but there's some sites in the C terminus of the alpha subunit. But what also could be happening is that when you have potent PKA activity in smooth muscle cells, you have increase in ryanodine receptor activity as well. And in smooth muscle cells, ryanodine receptors are key to activating these BK channels. So we're sort of agnostic about what's going on. I would guess it's a combination of those two things if they're actually occurring in this tissue. But yeah, we didn't go into the nuts and bolts of PKA activation of the BK channel, but that's the presumption that we have.
While we wait for the next question, I wanted to ask you, has anyone looked at I'm sure people have done these studies of like protein expression in the urothelium. Like, is there anything
Yeah, I mean,
gleaned from that?
yeah, there are protein expression less so. I mean, you're sort of subject to antibody performance, things like that. But there's not a lot in the bladder on like detailed proteomics. There are more groups starting to do sequencing, single cell RNA sequencing, and I think that's providing a lot of interesting hypothesis for what the urothelial cells may be doing based on this type of genes that they're expressing. Um, but it's an area, ripe for discovery. I think it's just a few groups have done sequencing within the bladder.
Exciting.
Yeah.
Next question from Zygmunt Kaltitsky. Is bladder affected when you are in the mountains?
Yeah, it is actually. I mean, I don't think we wrote this in the paper, but there's several studies showing when adaptation to high altitude, one of the symptoms is uh, I think it's a less frequent voiding, which is interesting and kind of goes in line with this hypoxic effect.
We don't have any more questions, then join me in thanking Nicholas for a great talk.
Oh yeah, thank you very much for the invitation.
Very welcome. And thank
Thank
you.
you.